Okay, guys, so we're going to do a fun experiment right now. So as promised, we're going to take some of your cells from your cheek, and we're going to do PCR and see what alleles you have.
Hi, John.
Hey, Bob.
You guys ready to go?
Yeah, actually, we already did a set because it's going down to UCLA by FedEx right now.
Oh, wonderful. Great. Thought Ryan be in the lab trying to do all that stuff.
Well, the kit didn't work, so we're trying to troubleshoot it, but just in case, we're sending you samples too.
Kit didn't work. I wonder if our kit's going to work.
It did work.
It did?
Yeah.
Great. That means when Ingrid sent it up, she didn't send it the fastest way, so it probably had some problems with it.
I don't know what happened.
She's blushing now, but it was a matter of contention. Okay, go ahead.
Okay, so if everyone can take your swab, and Daisy's going to demonstrate this, and take a pen and write your name legibly on it. Pen.
You have to write your first name, your last name, and write it very clearly. Write it very, very clearly.
It says sample ID on the white label on your tube, so write it in that area. It's not imperative that it's in the
Are you using a microphone or you're not?
It's my voice projects to the microphone. Kelly's holding because I'm loud.
Okay.
All right, you all set? So twist open the container, like Daisy, pull apart.
Daisy, stand up.
All right. Everyone figure out how to open? Just twist in the center, pull apart. All right, then take your swab. Oh, sorry, my bad. So rinse first. Okay, so put it back. And just open your water and just rinse out your mouth for like 10 seconds.
Rinse all that junk out of your mouth that you've been
We want a pure sample of just
munching
your cells,
on and
not your lunch.
your boyfriend and girlfriend's saliva, so we don't get a mix here.
Okay, so just for like 10 seconds.
Swirl it around.
Yes.
Did you guys do it yet?
Yeah, did it yesterday.
Where's your sample? On the gel already?
Min ran it already.
Okay, good. You guys ready?
Okay.
Okay, get it out of your mouth. Don't spit it on the ground.
Okay, so everyone ready? Take your swab again, open it up, and then just scrape the side of your cheek like 10 or 15 times, really hard.
Got to get a lot of cells on there because we've got to be able to get some DNA.
10 or 15 times on each
Look at
side.
John. John's doing it.
Yep.
Dora's doing it. Faria's not doing it. She's reading her notes.
Okay, and once you're done, you can put it back in the container.
Don't rub the side of the container. Put it in straight like you're threading a needle.
And once you're done, pass them to the right, and we'll collect them and do the PCR, and you guys can see the results later.
Okay. Alan McEwen, just a summary. I think, again, as I said, many of these speakers are not used to getting up in front of a class, so they're not going to hype you like I do and razz you like I'm going to do because they're just not that comfortable. But on the other hand, Alan had a lot of important things to say. And I think he had a lot of insight into not simply this GMO issue, but really critical thinking about any issue. You know, where's the meat anytime anyone says anything to you? Where's the evidence? Why do you think that? What's your hypothesis? Where's the data? And I think that's really the critical issue. And thinking about, as he said, alternatives. You know, oh, we can't use GMOs to prevent insects from infecting our fields because it's going to have a problem on the ecology. Well, what about pesticides? I mean, you have to think about alternatives. You know, if you do it one way and you don't want to do it that way, what's the alternative? Is that better? Is it worse? Is it the same? How do you mitigate these things? And I think that's really the key. The key is to be able to, it's not so important how you come down on the issue. What's important is how you think the issue out and come to a reasoned decision. And I think all of you need to work on that as much as you possibly can because it's a lifelong effort. Okay, this afternoon, after you leave today, maybe earlier or later, we'll see, you can pick up your bacterial experiment key and the key for the take-home, for the double helix essay. So those keys, and you should be getting those back. So Tiger, putting the microphone on your face, what was the hypothesis that you constructed for the bacterial experiment? Get her in the face.
My hypothesis was that each of the 3 vials contained bacteria that either had or did not have a plasmid.
How do you know that they contain bacteria from your experiment? From the results of the experiment?
Well, because what was left on the agar plates obviously grew. And
so there was some living thing in there, correct? You agree,
there.
It wasn't dust from the air, right?
Right.
And you knew it was living because of what reason?
Because with giving it several days to grow on the plate, it did
grow. So something grew.
Yes.
So it had to be living.
Yes.
Some creepy thing that, did you open it up and smell it or anything?
It leaked all over me. Yeah.
Did it smell nice?
Delicious.
Yeah, okay. Very familiar smell, not very pleasant. So then what was your hypothesis for the different results? Other than the fact that Daisy blurted it out in class. But anyways, what was your hypothesis for that?
I hypothesized that vial A, the bacteria didn't have any plasmid.
Okay, and because of what results?
Because, well, on the first plate...
You sound like Obama now. Well...
On the first plate, it was able to grow, and that was part of my hypothesis, too, that the first plate didn't have any antibiotic on it. So if bacteria didn't have any resistance gene, then it would be able to grow there, but not on the other 2 plates.
Great. And so keep going.
And so the first plate didn't have any antibiotic, and the second plate had one kind of antibiotic that B was resistant to.
Could have been more kinds, but
yeah,
it could have been 100 different kinds of antibiotics.
Some sort of antibiotic that vial B bacteria was resistant to, because it was able to grow on it.
So it was an antibiotic that B was resistant to, but there could have been 7,000 other antibiotics in there that had nothing to do with B. Do you agree?
Yes.
Or A. So what was your final hypothesis for your final result?
Well, bacteria C, I believed, had a plasmid that contained resistance to 2 different antibiotics. Or more.
You could have been
1,000.
15. Yeah, exactly. Yeah. It's only the 2 you were concerned with, correct?
Right, correct.
Okay, great. Dora, do you agree with that?
Yeah, I agree.
You agree? Is that what you put down on your results of your experiment there?
Pretty much.
Uh-huh. It's not so easy when you're working in a black box and have to come up with a reason for something when you don't have any frame of reference, isn't it?
Yeah.
Yeah, think about all the things that you've been reading in every book. Every sentence was from something like that. Nothing you read in any of your science books wasn't put there without someone doing some experiment like that and coming up with some hypothesis. Okay, let's turn to the double. What were the scientific, Faria, what were the scientific clues that led Watson and Crick to propose, what were some of them that led Watson and Crick to propose their double helical model? And from my spies tell me that this was something you had a little bit of difficulty with. Not you specifically, but you collectively. You know, if I was taking that assignment, I would have read the book, which I loved, but I would have gone online because we have webcasts, and the film was webcasted online, and I would have watched it again. Would have been a nice way to sort of look at all those things. In any case, Faria, you have your hand over your mouth.
So from the book that they knew that it was double helix, and
how did they know it was a double helix? Okay, what's going on with that?
Because due to the X-ray.
Okay, so they had X-ray. So they didn't know it was one of the clues was from the X-ray. knowing that it was a double helix wasn't a clue, that was a result.
Yeah.
Okay? So they had the X-rays. They had that cross. Remember the cross that Crick told Watson about? What was another clue, Anna? You have a microphone?
Avery's experiment?
How did the Avery experiment lead them to believe that get the double helical structure? I mean, Avery experiment certainly was important in one of the things allowing them to think that DNA was the genetic material. But how did that help them solve the double helix? Are you reading or are you thinking?
I'm thinking.
Okay. Do you know? I don't think the Avery experiment would have helped them. Michelle? Look at her eyes get bulging big. Woo! What were some of the clues that Watson? So definitely the X-ray diffraction of the cross, the B structure. What else? What are you reading here? 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 What? 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 These guys got the keys and they're just reading it off. What do you think about that? Okay, Max, what were some of the clues? You don't have that thing in front of you.
Chargaff ratios?
Yeah, Chargaff's rules, no question about that. So the B structure, X-ray diffraction, Chargaff's rules. What else? Jessica. What? Talking to the microphone. Come on, get used to it. I don't think it's on.
Yeah.
So what were some other clues? Come on, this is, you watched a movie, read a book, wrote a little report.
DNA it has like hereditary material.
What?
It's hereditary.
That has nothing to do with the double helical model. They knew that. Okay, that has nothing to do with how they solved the double helix. So they had X-ray pictures, they had Chargaff's rules. What else did they have? Remember, you're trying to solve a problem. You're in a black box. You don't know anything. You're trying to come up with something. Elaine. Come on, Jessica.
They knew that DNA had a phosphate sugar backbone.
From what data? They knew, they just made it up? How did they know that? From what pieces of evidence?
From the X-rays?
Yeah, from the X-rays. So they knew from the X-rays that it was a helix. They knew from the X-rays that it had a sugar phosphate backbone. What else did they know from the X-ray?
That the backbone was on the outside?
Well, that's implicit.
Oh, okay.
Okay. What else did they know from the X-ray? Remember in the movie and in the book, Crick was showing Watson about, the ladder and how far apart the rungs on the ladder were. And he said, and it goes around, and this is the pitch. And
oh, they knew the measurements.
Yeah, they knew the measurements. They knew how wide it was, and they knew how many nucleotides, were spaced, what the spacing was of the nucleotides. They knew how many nucleotides per turn of the helix. What else? What else? What was one other fact that was very important? Dora, do you remember?
Would it be the water content?
Not so much, but that's a minor thing. Yes.
The keto
Nora.
form of the bases?
Keto form of the bases, absolutely. The chemistry of the bases. Remember, the guy said, no, it's not like this. Crossed it out in the movie. It's like this from the chemistry. Remember? Yeah, it has a certain chemical form. It's not important to know what keto and enol is, but that's clear. And then there's one other important fact. Katie?
DNA, would it be that DNA needs a method of replication?
No, because that doesn't tell them very much about the structure. Because the structure, even if it was single-stranded, you could come up with a model. So right? I mean, whatever the structure was, you could come up with a model for that. So what was the one other thing? Rows. Rows arose, yeah.
Wasn't it also like at the very beginning of the movie, even they said that DNA had a regular crystalline structure?
Well, that was implicit from the X-ray diffraction.
Well,
that doesn't provide any, I mean, that's, yeah.
Well, but then in the book, Crick or
So
Watson
what was
saying...
another fact? What was another very crucial fact? You've got dimensions, you've got backbone, you've got Chargaffs, you've got keto, Eden.
This is also from the X-ray, but Wilkins deduced that there must be at least 2, 3, or 4 strands of DNA.
Could be, but one other very important fact, one of the most crucial facts, Samantha.
Was it the fact that one of the strands was the 5' end was matched up with the opposite side, like one of the strands was running down?
Anti-parallel nature,
Anti-parallel
C2,
nature.
remember? Crick said, C2, she missed it. The C2 group, they're anti-parallel to each other. She didn't see it, didn't see C2. So that was critical. And in reality, as you can see on the screen, all of the above. There wasn't one of those things by themselves would have allowed them to propose a really adequate model for the DNA double helix. It was really all of the above. And so my spies said that there were some difficulties with that part of the little report. So that's why I wanted to go over it and just sort of, if you go through it logically, I think it becomes really clear. Okay. Let me move on, or I should say move back, and then I will move on. And let's just go back to where I didn't want to save it. Not what I wanted to do. Didn't want to crash it either, which I probably just did. Okay, so let's move forward. We're going to move backwards and we're going to move forward. So last time I was talking about, and I summarized some stuff on here for you, but we don't need to go over it. Last time we were talking about how do genes work and what do they do. And I talked to you about a couple of things, and that was about what a gene is and the anatomy of a gene in 5', 3', sense, antisense. Ryan's there, template, transcribed, all of those things. And then I talked about DNA replication and how that worked. And I talked about semi-conservative DNA replication. And I talked about then how you do DNA synthesis in the test tube, the polymerase chain reaction. And then we saw that wonderful movie by that Nightline thing about Mullis. And then I talked about applications of PCR. And then I talked about mutations and how mutations arise. And we've talked about genetic variability, so I discussed that. And then I talked a little bit about fast forwarding about GMOs and how you think about genes and how they perform. But I really haven't said very much about how the gene gives rise to the phenotype, even though you all know how that happens. But let me just review for you very quickly, because it's very important that I remind you of what I said last week about the collinearity between the DNA sequence in the gene, the messenger RNA sequence, and the protein sequence. And here it is. And so we have the DNA double helix. We have 5 to 3, 3 to 5. Remember, the sense strand is always defined as 5 to 3, and that's how we define a gene. The antisense strand, or the transcribed strand, is transcribed into RNA. We have it anti-parallel, 5 to 3. And then we have that translated and made into a protein in the cytoplasm of the cell. And notice I haven't said very much about proteins, but they consist of amino acids. That's the unit of the protein. That's comparable to the nucleotide and nucleic acid. They are linked together by bonds called peptide bonds, not phosphodiester bonds. And the protein has a beginning and an end. The beginning is called N, and the end is called C. It's not important why it's called N and why it's called C. It's slightly different molecules. But the point here is is that the beginning specifies the order, and there's also polarity in the protein because of polarity in the amino acid, and a unique amino acid gives rise to a unique protein, and in this case, insulin. So it's a beautiful, beautiful, elegant way in which your cells tells the cell what protein to make. And of course, last time we talked about the switches that turn that on and turn that off in very specific ways. Now, all of that happens, as you know, because there's this thing called the genetic code. Would you actually believe, Chris, and I know you're going to say yes, you just look at the color of my hair, that when I was sitting in a class like you are now at your exact age, the genetic code wasn't even known. It wasn't until I was working on my PhD in graduate school that the genetic code was cracked.
Yes,
so my scientific career is paralleled to the discovery of the genetic code, genetic engineering, genomics, and sequencing. Isn't that exciting? All of these major discoveries.
It's pretty cool.
I don't know if it's cool or not, but it's just a fact. I'm sure
Pretty
you're going to be able to say the same thing when you get to be my age. But the point here is that we have the messenger RNA translated into a protein. And as I said again, remind you, protein has a beginning and an end. And notice, very importantly, that the 5' end of the gene and the 5' end of the messenger RNA correlates with this N end of the protein. And that's a very important thing. And that happens because there are these codons, the genetic code, which is universal, as you know, because you'd never be able to get the glowfish protein made in a plant or in a bacteria or in anything if the genetic code wasn't universal. And that was a really big deal to demonstrate what the genetic code was. Many Nobel Prizes were awarded in the late 60s, early 70s for the people that uncovered the genetic code. And I must say, Crick was involved in this as well. But the point here is that the genetic code has some properties. It's universal, as you'd expect from the experiments. And you should be able to design an experiment to show that the genetic code is universal. It's a triplet code, and that's because the only way you can get 20 different amino acids, and there are 20 different amino acids inside your cells, is to have 3 nucleotides. Because the only way you can get enough different combinations of nucleotides is to have 3. If you only have 2 nucleotides in your code, you can only get 16 different combinations. If you have 3 nucleotides, you have 64 different combinations. And because of that, the genetic code is said to be degenerate. That means there's, and this is very important for something that I'm going to talk about today. You can see that there's many more codons for certain amino acids. So you can get to a certain amino acid in more ways than one. And that has some consequences evolutionarily. It has some consequences because it might mean that you can mutate. Notice GCA, GCG, GCT, GCC. You can change that last codon nucleotide, and it doesn't change the amino acid at all. So that means some mutations in genes won't lead to a phenotype. They'll lead to a change, a polymorphism, a difference at the DNA level, but they're not going to change the phenotype. So keep that in mind. But the point here is that you have a triplet code, you have a degenerate code, and most importantly, you have punctuation. The signals that you learned about at the protein level, so to speak, at the messenger RNA level in discussion. You have a nucleotide, a codon rather, that is the start of translation, and you have a codon which is the stop of translation. So when the proteins are being made, when the messenger RNA is being translated into protein, the cell knows to start translation at this codon. It's AUG, because remember there's U in the RNA rather than T. RNAs have uracil rather than thymine. So this is the start. It's always a methionine, always in general. It's always AUG, and notice that there's stops, UAA, UAG, UGA. And so the messenger RNA has a beginning and an end, and the beginning is a particular codon that says start translation. And notice the end, there's no amino acid that correlates with that end stop codon. And that's why protein synthesis stops. Okay? So universal, degenerate, punctuation, and triplet. And that was uncovered in the early 60s but is very important. So that means, as you know from your BLAST, if you know essentially the gene sequence, if you know the gene sequence, you know the protein sequence automatically from the genetic code. But, Mike, right? But if you know the protein sequence, you don't necessarily know the gene sequence. Is that correct?
That's correct.
Why is that? Where's your microphone?
Because, as you said, the genetic code is degenerate, so multiple.
Yeah, exactly. And that's going to be very important when we talk about patents and intellectual property, because that means that you can't go from the protein sequence to the nucleotide sequence because of degeneracy of the code. And that has a huge history in the patent realm, as I'll talk about in a few weeks. And that'll be important for what I'm going to talk about today. And so then clearly, if you get a change in a codon that leads to the codon specifying a different amino acid, not a change in a codon that might be neutral, that doesn't change the amino acid that the codon specifies. But if you have a change in a codon, like you do here in the globin gene, that changes this nucleotide to that nucleotide via mutation, and if it changes the amino acid, in this case from glutamic acid to valine, notice that you can change the structure of the protein. And so notice that the protein has a shape. All protein has structure. Proteins work because they have a structure. And notice in the case of sickle cell anemia, that when you change this one nucleotide right here, you change this one amino acid, glutamic acid to valine, what happens is that the globin proteins become very long. And when they become very long, let's just say they don't fit in the blood cells as well. And when they don't fit in the blood cells as well, they cause sickling, and they don't bind oxygen as well as the normal type. And so it has devastating consequences, as you can see here, for individuals that have the sickle cell anemia phenotype. So you change the codon. In many cases, you change the amino acid. You change the amino acid. You change the structure of the protein. You change the structure of the protein doesn't function the way it's supposed to. If it doesn't function the way it's supposed to, then you get a phenotypic change. Go back to you, Mike. Remember I threw you the purple cabbage? Remember the purple and the white cabbage? Now give me a molecular explanation for how that occurred. So if we have the purple gene, there's the purple gene. What might it do?
So the purple gene.
What might it encode?
We determined, I believe, that was the original.
Well, what do you think? Just hypothesize.
Okay. I hypothesized that at some point there was a mutation.
But what do you think the purple gene does? Let's start there in the cell.
It expresses pigment
acceptance. It makes a pigment. Mm-hmm. And the purple gene encodes a protein. Is that correct?
Correct.
What kind of protein do you think that might be? You think that protein might be involved in a reaction in the cell?
No.
You don't think so? What do you think might it be? You think it might be a pigment protein? Yeah. That's possible. It's not really, but let's just call it a pigment protein.
Fair enough.
That's fine, fair hypothesis. Dora, do you agree with that? That this purple could be a pigment protein, or it could be an enzyme that converts chemicals to a purple color.
Exactly.
Could be either or, okay? You don't know it until you clone it and find out. So now, the purple gene then changed to some green gene, right?
Uh-huh.
What are the different forms of that gene called?
Different phenotypes.
What are the different forms of that
Alleles.
same allele? Okay, they're alleles. And how did the purple gene change into the green gene?
At some point, a mutation. In
what kind of mutation? Tell me the different kinds.
There's deletion.
Okay, a deletion.
Insertion.
Insertion.
And general mutation.
What's a general mutation?
The word I'm looking for is.
What's a general mutation?
One in which you get a change of a certain.
Okay, a change, base pair change. So we have deletions, insertions, base pair change. Can you think of anything else?
Yeah, you can have entire sections that.
Maybe entire sections switched or something, or taken out, right? Yeah. Okay, good. And so then we have this green gene. What's your hypothesis for why we get the green cabbage?
At some point in the history of this cabbage, there was a mutation from the purple gene to the green gene, or for the protein that coded for that change, and it was advantageous
So how come this thing gives green instead of purple?
Because
give me an explanation.
The pigment...
Give me an explanation.
So you have a shift
What happens to the protein?
One of the proteins, we'll go back to the base pairs, one of them changed.
So the base pair changed, right?
Some
messenger RNA changed.
Mm-hmm.
How come the protein doesn't produce the pigment anymore?
Because it now has a different acid that's coming in.
Amino acid. Yeah. Does it have the same shape as the normal?
No, the
has a different shape. And so now let's say whatever made it a purple pigment protein, it can't bind something or do something, correct?
Yeah, exactly.
Okay. What you've just seen is a prelude to your oral exam. Every single thing that you say, you have to know what it is. And then when you say what that is, you have to know what you said, and it keeps going back and back and back conceptually. You say DNA, what's the structure of DNA? You say nucleotide, what's a nucleotide? You say phosphate, what's phosphate? Say complementary bases, what's complementary bases? You say double helix, how does it replicate? What is replication? Semi-conservative. What does semi-conservative mean? You need a template goes to one strand goes into each of the daughter DNA molecules. What do you need for DNA synthesis? Primer, template, nucleotides. How do you do that in a test tube? What's it called? PCR. What's PCR? How does PCR work? So the way in which you study for this exam is you make connections just like you did. Remember when you were a little kid, you had all those little dots and you followed them. And so anything you say, you just keep going. And you keep answering your own questions, and it's a really good mental exercise for anything, whether you're going to go into the law or whether you're going to go into science and where you go into anything. Where's my little cap? Can you cover that for me? Okay, so that's how we get, that's really good, Mike. Thank you very much. So now we have, and I'm just reviewing here before I move on. That's why I said that I'm just going back a little bit before I go forward. We know that we have universal processes in bacteria and in eukaryotes. Universal genetic code, universal processes of protein synthesis, universal processes of DNA replication. We know that the DNA structure is the same. Dora, how do we know that? What is the evidence for the fact that the DNA to RNA to protein processes in a bacteria are the same as a eukaryote? Please design an experiment for me, something that you've done in discussion in which proves that to be the case.
We can use the recombinant DNA technique by taking a gene from one bacteria or eukaryote and inserting it into a different
I agree with
bacteria.
you, but that only says one thing. Does that say that the expression processes are the same from a eukaryote to a bacteria, or does it say maybe just the structure of the DNA is the same? Okay, so what was the experiment that showed that the expression processes are the same?
So you can take
give me a specific example from discussion.
Like the gene from the jellyfish,
We didn't
and then
have
you
that
can
in
insert it
discussion.
into fish.
I mean, that's a good example, but what about from discussion? What was the example of a eukaryotic gene that you stuck in a bacteria, and you had a long discussion about it? What do you think? You want to caucus
How about the genes from bacteria and insert into plants?
No. What about Faria? Want to help out? You want a caucus here? Want to have a little caucus? What about second week? What do diabetics use?
Insulin.
Yeah. What about that experiment? What was that experiment?
That only gets expressed in like a certain area of the body?
Oh, yeah.
What was that?
It's insulin's created in a certain part in the liver or something, or pancreas, and
how does that experiment show? How does that experiment show, Max, that a eukaryotic gene can be active in a bacteria?
Both.
Tell Dora.
Oh, they both. They both expressed the same. They both expressed it, so both made the protein.
What's both? I don't know both.
The pancreatic.
So the pancreas makes insulin.
Yeah, the pancreas makes insulin.
And then when you stuck it in the bacteria,
they started making insulin.
Yeah, so that shows that the expression processes in general are conserved, which means the genetic code is conserved, transcription, translation. But very quickly, and I think you all know that because you had it in week 2 of discussion, and this is a review, there's slight differences. And here's the major difference. Prokaryotes don't have a nucleus. Isn't that right, Lauren? You're taking LS2 now, right? No, you're not. Thought you were. Who's taking? Someone's taking LS2. There you go. They don't have a nucleus, correct? Pardon? You're positive. Yeah, you're positive. Okay, you're right. The point here is they don't have a nucleus. So that means transcription and translation goes hand in hand in bacteria and other kind of prokaryotic cells. But eukaryotes have this big, fat brain center called the nucleus, which means that genes are in the nucleus. They're segregated from the cytoplasm. It means that genes have to be transcribed in the nucleus. The RNA that's made has to be transported out of the nucleus and into the cytoplasm because the protein synthetic machinery occurs in the cytoplasm. So again, prokaryotes have a chromosome, and we know that. It's circular. Transcription occurs, translation occurs. There's a cytoplasm. All the protein synthetic apparatus is in the cytoplasm. Same with eukaryotes, but the genes are segregated from the cytoplasm by this nucleus. So that means that prokaryotes don't have to transport their messenger RNAs out of a compartment into another compartment to get proteins made. But eukaryotes have to transport the RNA out of the nucleus into the cytoplasm in order to have proteins made. Thomas, give me a hypothesis for the structure of a nucleus. How do these RNAs get out of the nucleus?
They travel outside. There's a chemical process that allows them to escape.
How do you? What's the simplest way in which you think they get out? You think these RNAs are small?
Yeah.
You think they're small?
Yes.
What's the average size of an RNA? How many nucleotides?
Fairly small, 100. 100, I would guess.
100. What's the average size of a gene in the human genome? Do you know yet?
The human genome?
Yeah. What's the average size of a gene?
Rather large, thousands possibly.
Thousands or tens of thousands?
Tens of thousands.
So how many nucleotides? If you have a gene that's 10,000 bases in length and there's no introns, what's the size of the messenger RNA?
The same length as the DNA.
Yeah. So. You think it's small or you think it's big?
Big.
How do you think it gets out of the nucleus?
Honestly, I don't know.
What do you think? Make a hypothesis. That's not an acceptable answer.
There's a chemical process that allows them to escape the nucleus.
You think it's a chemical process?
It would be my hypothesis, yes.
How many messages do you think are coming out of the nucleus at one time? 1, 10, thousands?
Thousands, tens of
exactly.
thousands.
So you think that there's tens of thousands of chemical processes going on?
At the same time.
How do you get into this room?
I walked in.
How do you get out?
Through a door.
How do you think the messages get out of the nucleus?
Through a door in the
exactly.
nucleus.
So there's little holes in the membrane of the nucleus and allows these things to get out and go into the cytoplasm. And it's a pretty elaborate process. But the point here is they got to get out. Now, the one thing that I don't want to get into, because it's the source of a whole course, is that eukaryotic genes have these things called introns. And this was a major surprise. Once a eukaryotic gene was cloned in a plasmid, and they were able to study the structure of the gene. Remember, before the first article that you had for the first week of class, no one could look at a gene and know what its structure was. So the first time someone cloned a eukaryotic gene, they looked and they saw, lo and behold, they looked and they saw there were bits of pieces in that gene that weren't represented. Can you put that down, Mike? That weren't represented in the messenger RNA, meaning they had a clone of the gene, they had a clone of the cDNA, they compared the gene sequence to the cDNA sequence, and they found little bits of sequences that were not in the messenger RNA or the cDNA that were in the gene, and they called those things introns. It just so happened that I was at the meeting in which those introns were first reported back in 1976, and no one believed it. And one of the reasons why no one believed it is no one believed that there was things inside of a gene that had nothing to do with what the gene was doing. And in the early days, people thought that this was an artifact of cloning. And it wasn't until a bunch of other experiments were carried out that it was demonstrated that it wasn't an artifact, that it was in fact a reality. So that means, Tiger, what's the problem that the gene has in specifying the protein if it has these bits of pieces in them that don't code for the right amino acids in the protein? What's the problem?
They have to be removed first?
Yeah, if they weren't removed, what would happen?
You would start getting proteins that you didn't need?
Yeah, you'd start, well, not proteins you don't need. Go ahead.
You would get codes for amino acids that didn't make up the protein that you
Yeah, and
wanted?
would the protein function?
No.
Yeah, so the protein would have amino acids in them that it didn't want, and the structure of the protein would be different. You buy that, Thomas. So what has to happen then?
They have to remove the introns?
Yeah, and so it has to remove them by a process after transcription called a post-transcriptional processing. And that was something which was really remarkable and also won a Nobel Prize, of which people were very, very surprised. So again, that was one of the first major surprises about genes after cloning was invented in the 70s, that genes had bits, eukaryotic genes. Prokaryotic genes, for the most part, don't have these extraneous bits. So that means that we have to have a splicing reaction. These bits have to be taken out of the RNA. Derek, are they taken out of the gene, or are they taken out of the transcript of the gene?
The transcript.
Okay, what would happen if they were taken out of the gene?
Then the gene would be changed.
Exactly. Can you think? Do you think there's any situation in which things are taken out of genes in your chromosomes? Do you think anything's ever taken out of your genes in your chromosomes during your lifespan, meaning from conception till the way you are now sitting here? Yes or no?
Yes.
You know, do you do? What's the example? You mean bits of things are taken out of my genes and your genes as we develop?
Mutations, yeah.
Okay.
Unfortunate.
Could be somatic mutations. But there is one example, actually, in which things are taken out of your genes.
Transposons?
Not really, but it's in making the antibodies that you make, but I'm not going to get into that. There's one, for the most part, things aren't taken out of your genes, but they are taken out of these transcripts. Now, give me a hypothesis, Derek, for how the nucleus knows to grab these things exactly at the boundaries of those introns and stitch them together. Does it just do it magically, or what does it depend upon? What's the one thing that always guides everything in biology?
Other genes that make
Other
proteins.
genes make other proteins. Okay, so that's true. There has to be other proteins to do this. But what guides that process? What happens everything in biology, in genetics?
The DNA.
Give me a simple hypothesis for how that happens. We have a dilemma here. Now the going's starting to get a little bit tougher, because now you got to start synthesizing all the things that you're learning. So it gets ramped up just a little bit more, okay? So what would be your hypothesis? Eric, I mean. What would be your hypothesis? How does this process, what guides this process? Starts with an S.
I was going to say regulatory genes, but...
Well, I mean, there are genes that make proteins that do this. I mean, it doesn't happen by itself, although in some cases it can. But
Maybe.
What guides it? How do you know that there's an intron here and an intron here that has to be taken out?
Maybe there are certain repetitive sequences.
What did you just say?
Repetitive sequences.
What guides everything in genetics?
Sequence of the
sequence of
nucleotides.
something. So do you think that there might be specific sequences there?
Yeah.
Is there a specific sequence of an origin replication?
Yeah.
Is there a specific sequence of a promoter?
Yeah.
Is there a specific sequence for the start of translation?
Yeah.
Is there a specific sequence for the stop of translation?
Yeah.
Is there a specific sequence for the start of transcription?
For
Specific
everything.
sequence for the end of transcription?
Yep.
Is there a specific sequence for a particular gene that makes a specific protein?
Definitely.
Is there a specific sequence of a protein at the amino acid level?
Yep.
So what might guide this process?
Sequence.
Exactly. And there it is. So again, there's specific sequences that guide this process and cause this intron to be lashed out in the nucleus by this processing and splicing. And of course, proteins actually do take part in this process. It's fairly complicated. But the point here is that there's sequences of everything. Now, let me come back to that. So that means that genes and the way in which they function, Chris, have a modular organization. Right? What are the consequences of that, Brian, for genetic engineering? You with me, Dora?
Yeah.
What are the consequences of the modular organization of a gene for genetic engineering? Do you ever play with Legos?
Yeah.
So what's the consequences?
It has to be like built a specific way.
So what are the consequences for genetic engineering? What does that mean in a big sense, in a big world?
We have to be able to look at the sequences and look for...
You look for the sequences, and you have to look for these modules. But I gave you these modules in this Lego kit. What does that mean? Let's say I give you the module for every gene that was ever constructed on the face of this earth by nature. You have the coding sequences, you have the starts, the stops, everything. What does that mean with respect to genetic engineering?
I have to know how to put them together. And
well, how do you put them together? You know how to put them together because you had a quiz this week that told you how to put them together, right? By splicing and putting in plasmids and stuff. So what does that mean, Albert, for genetic engineering? What's the consequences of this?
That we can combine different genes.
Yeah, you can combine different genes. What about the glowfish gene? What about insulin in bacteria? The consequences of this for genetic engineering is if you have the units, you can do anything. There are no limitations. You have a vector, you have restriction enzymes, you have a cell in which the vector can replicate. You can do anything. You can put all these things together. You can create a start of transcription. You can put a novel switch in front of that start of transcription to get something active in an eye or a head or wherever. You can put a stop on. You can put any gene in between. There's no limitation. Derek, do you see that? Tiger, do you see that? That's the point I was trying to make the first week of class. There are no limits to genetic engineering. None. And what would be the consequence of sequencing and understanding the genome of every major organism on the face of this earth is that you'll have all of this material at your hand. And you can just start mixing and matching and doing anything you want once you know what they do. No limitations. And so that's really the point. And the point then is that I need you to think about, and I talked about this, and this is the penultimate thing that I'm going to say about this topic, is that when you're thinking about GMOs, and I've said this, whether it's animals, whether it's humans, whether it's plants, whether it's bacteria, and I've said this many, many, many times, and I'm going to say it again, it is not hocus pocus. You're dealing with very specific sequences, you're dealing with very specific processes, and you're dealing with a very precise engineering process of which chance is completely eliminated. I'm saying that emphatically. There is no chance in any of this. You know what the sequence of the ingredients are that you're using. You know what the sequence of the protein is. You know what the sequence of the amino acid is. There is no mysteries. You know every single thing that you're engineering at the molecular level down to the atom. So when someone says, well, you know, this can do this and create monsters and do all these other kinds of things. Well, Elaine, how come your genes don't create monsters inside your cells? It's no different than a gene I could engineer in you. The point is, how come your genes aren't just mutating crazily all the time and bombarding your other genes so that your cells become monsters? It doesn't happen. When you engineer something, it's the same process. The structure of the DNA molecule is the same. The replication is the same. The transcription process is the same. Protein synthesis is the same. The modules are the same. The molecules are the same. The atoms are the same. There's absolutely nothing left to chance. So what's more precise, Kathy? Doing what Karpachenko did, taking a radish and a cabbage and coming up with a plant that had the roots of the cabbage and the head of the, right? Or doing something very precise like this.
Doing something.
Yeah, I led you into that, but the point is that it's a very precise process. And since 1973, when genetic engineering was invented, I would venture to say, and maybe John, you can agree, there have been tens, oh, John's gone. Anyways, there's been tens of millions of genetic engineering experiments in labs all over the world. Now, I'm not aware of anything that's ever happened. I'm not aware of anything that happened to the children that have been engineered with adenosine deaminase genes, except that they have a healthy life. I'm not aware of the goats that have genes in them that are making therapeutic proteins that can help your grandparents stay alive if they have heart problems. I'm not aware of anything that's happened in all of the GMOs that have been released into the environment over the last decade all over the world. I'm not aware of a sneeze, a cold, anything. And why shouldn't it be that way? Because we're just dealing with science and genes. It's not a mystery. And when you're thinking about these issues, I don't care about this stuff. Ben, I don't really care if you come back to me 10 years from now and you say, I can't remember one damn thing you taught me. I don't care. But I care that you can think this issue out like this. That's what I care about. That's the most important thing to me. That you can think it out logically and critically and come to a conclusion. And I don't care what your conclusion is, but you can give cogent arguments along the way. And you won't say, I don't know, or I don't know what's happening, that you can give a very logical, because as I said the first day of class, and I'm going to end this little discussion, we are living in the world of anti-science. We still live in a world where school boards want to ban teaching of evolution and still think that it's a theory, which it's not. It's a fact. I mean, just look at genetic engineering. We still live in a world where people think that if you get vaccinated, you could come down with all these different kinds of things, like autism, even though there's been millions of scientific studies that have said that's not the case. The point here is, in an anti-scientific world, where people still think that plant GMOs are going to create Frankensteins, one needs to be able to use logical arguments one way or the other that are based on good and sound science. And that's why I'm pounding this into you. You understand these processes now. We don't need to go over them anymore. You understand the basis of this technology. You understand the basis of these genes. You understand their modular organization, how conceptually they can be turned on and turned off, and how they work and why they're universal. And therefore, you need to use that in constructing logical arguments, whether it's based on science, whether it's based on policy, whether it's based on whatever. Okay, let me move forward. Are there any questions on anything that I've said up to this particular moment? Samantha, my woman.
Okay, so just going back to the drawing behind you of the introns, and you were saying it's an artifact of cloning. What does that necessarily mean, an artifact of cloning?
Yeah, that means an artifact of cloning means that if you put a gene in a bacteria, maybe some of the processes of replicating the gene in the bacteria had extraneous bits of DNA stuck in the gene just because it was a mistake caused by replicating a eukaryotic gene in a bacterial cell. Remember, in the origins of genetic engineering, they didn't know that a eukaryotic gene could be replicated in a prokaryotic cell. Even though we knew that the general structure of the DNA was the same, but it could have been that there might have been little chemicals on the DNA in a bacteria that weren't on the DNA in a eukaryote or vice versa that caused something to go wrong in the replication process.
So this kind of transformation has been going on a lot longer than just human induced? transformation, where we're putting, you're saying these are artifacts of when we
No,
were...
I didn't say there were. I said that was a hypothesis.
Okay.
I said that was one of the explanations.
Okay.
I said people thought that it might have been an artifact of cloning, but it turned out that it wasn't. It was real.
Okay.
Okay? Thomas.
So for these...
Shoot.
Okay. So for these introns that we have on, are they just eliminated, or because the picture?
It depends upon where they're eliminated. Okay. Are they eliminated in the genes? No, but they're eliminated from the transcript, the primary transcript of the gene, and they're removed in the nucleus and they're degraded in the nucleus.
So they're separated from the RNA and then they're just sort of degraded back.
They're just not sort of. They're degraded back to nucleotides, and those nucleotides are then used as raw material for the transcription of new genes.
Okay.
Okay? Eden.
So the introns are also part of our evolution, right?
The introns arose during evolution.
So would it be possible to remove the splice sites and then look at the introns and sort of understand more about evolution of a species?
I think you have to be a little bit more specific about that.
Okay, so let's say you know an intron arose because there's a mutation and then the organism then used that gene to have a phenotype.
Yeah.
And so no longer uses that intron. But if a scientist wanted to understand what that intron was before the species evolved, could they remove it and then put that in and clone that intron?
Let me see if I can interpret your question.
Okay, sorry, it's
with
a
an
little
answer.
convoluted.
People have studied genes and their evolution, and people can find a conservation of introns in genes from related species, and those introns are in the same positions of the gene in related species, which means they evolved way back when, before those species split off from their common ancestors. Okay? But then you can find related genes, let's say, in prokaryotes that have some relatedness in terms of maybe function, which don't have those introns. And so one can then say that those introns arose after the prokaryotes and eukaryotes diverged at some point in evolution. And then those introns got into these genes, and there's various hypotheses for why they're there. Okay? And one of the hypotheses for why they're there is that in evolution, things move around a lot. I think in your reading you might have read about transposons or whatever. Things move around. And if things are moving around, and if they move into genes, they can have some serious consequences. So it's probably a consequence of, in a simplistic sense, of the eukaryote having events in the nucleus that it doesn't like and being able to deal with them so that it can still survive. And that's the simplest possible thing. But yes, you can use introns in a variety of ways. Yes, Julie.
Is the introns where the promoter and the activator and the repetitive sequences that you use for DNA for fingerprinting
Say that more slowly so I can grasp what you're saying, please.
The content of the introns, is that the promoter and the activator of a gene and the repetitive sequences that were used for DNA fingerprinting?
So what is the content of the intron? The content of the intron can be anything. It's just a sequence. It could be, it's not the promoter, but it could have, in some cases, rarely, it could have a regulatory sequence in it, but it's a rare situation when that happens. But let's just say it's just a DNA sequence. It doesn't specify a protein, it's just there. But it has things at the end of the intron which mark the beginning of the intron and the end of the intron so that the splicing reaction can occur in the nucleus. But it's just a sequence. And it's not the repetitive sequence that you're dealing with in your article or in your book that has to do with these variable number of tandem repeats. Those have nothing to do with introns. Okay? I know you're confused, but we'll get to that. Just look at it as a random sequence. It's just a sequence. It's just there.
Are introns the, I remember in one of the readings they were saying, I think it was introns that, depending on the type of cell, cut out different types?
Okay, good question. So I skipped that slide on purpose, but I'll go back to it. There it is. Let's say we have, who should I ask? I'll ask Zach. Let's say we have a cell and we know, and let's just say, and listen, Dora, because I may come back to you. Let's say we have a eukaryotic cell that has 75,000 different proteins. Okay? Different ones, meaning they do different things. And let's say we know from sequencing of the human genome that we only have 25,000 different genes. How can you get 75,000 different proteins from 25,000 genes? Theoretically, one gene, one protein. That's a real paradox, isn't it? So how do you think that might happen? What's your hypothesis?
You can order the genes in different...
I can't hear you.
You can put the genes in different orders?
We only have 25,000 genes in the genome. We can't change that number. It's fixed. It's been fixed since hominids were formed millions of years ago. So how can we get 75,000 different proteins when we have only 25,000 genes in our genome? What do you think? What's your hypothesis? Can't take them out and make different orders, Zach.
Could there be mutations?
Well, you mean that in one of your cells, I'm going to start mutating your genes to make 75,000 different proteins. Now, you know that that's not a good hypothesis. So let's come up with a good hypothesis. How do you think that might happen? What are the facts? You see, the reason why you get stuck with all of this stuff, Moore is giving me the evil eye. The reason why you get stuck with all of this stuff is that when I ask a question, you start saying, oh my God, and you just stab at something. But all of you know that's not logical thinking. In order to answer the question, you have to think about what you're dealing with. So if I say there's 25,000 genes, you're supposed to say to yourself, what's the structure of a gene? How are genes put together? And from that knowledge of what's the structure of a gene, you then come up with a hypothesis how maybe one gene can specify a couple of different proteins. And so you can't just say, oh my God, I don't know. But you do know. I know you know. Because right up here you see a gene. And it has all those introns in it. And you know that there's splicing inside the nucleus. And so the hypothesis could be that maybe there's different ways in which these RNAs can be put together. I mean, you all know about movies and editing movies. There's a million ways in your iMovie that you can splice together scenes or pictures. Why can't you splice together these little exons in different combinations in the nucleus? If you splice together exons in the nucleus sac in different combinations, changes the genetic code, changes the amino acid sequence of the protein. So you can get different proteins made from the same gene. And that's the reason. That's the answer. So in eukaryotes, you can have this thing called alternative splicing, which means you have one gene, you have one transcript that's transcribed, but then you can make different messenger RNAs by mixing and matching different exons and splicing them together in different combinations. And therefore, the sequence of the amino acids in the protein will be different for each of those proteins. Even though they came from the same gene, the sequence will be slightly different. And in this case, you can have a protein that functions in the skeletal muscle, a protein that functions in the smooth muscle, a protein that might not be functioning or function different in the liver, might be functioning different in the brain. And this is the way in which humans and other eukaryotes can make more than one protein from a given gene. And so, therefore, we're not limited by the number of genes we have, because if you have these exons and introns in genes, you can come together with different combinations. Is that logical, Tiger? And that's called alternative splicing, and it happens a lot. And again, that was a big surprise, too. And so it means that you have, what's happening is that as organisms evolve, what's important in evolution is not what's going on in the cell. What's important is what we look like. That's what That's what selection is, what we do. And it doesn't matter how we do it. If it offers an advantage, we can get there. And alternative splicing is one of the ways you can have more proteins for a limited number of genes. And so that solves a dilemma, Thomas. The dilemma is, how come, would you agree that a human is more complicated than a plant? But the plant has exactly the same number of genes. So how can we be so much more complicated with the same number of genes as a little plant? And it has a lot to do with this, and how many different proteins are made from the raw material that you have. But from a genetic engineering point of view, it also means that we can do a lot more and play and get all kinds of different things, but I'm not going to go into that.